is a tool, it can be applied in all the disciplines. ಅಂತ ಅವಾಗ ಸೆಂಟ್ರಲೈಸ್ ಮಾಡಿ ಅಂತ ಸೆಂಟ್ರಲೈಸ್ ಮಾಡಿದ್ರೆ ಎಲ್ಲರೂ ಯಾರು ಬೇಕೋ ಮಾಡ್ಬೋದು. ಆರು ಏನು ಗ್ರಾನೈಟ್ ಸ್ಲ್ಯಾಬ್ಸ್, ಆರು ಗ್ರಾನೈಟ್ ಸ್ಲ್ಯಾಬ್ಸ್ ಮೆಸ್ಸಿಗೆ ಟೇಬಲ್ ಮಾಡ್ಬೇಕು.
Good morning, Dr. Paul Raj. Can you hear me, sir?
I'm audible. నా ఫోన్ లిఫ్ట్ నుంచి Okay. ಯಾರೋ ಅಡ್ತಿರೋ ಬಂದ್ಬಿಟ್ಟು.
Good morning, sir. Can you hear
Good
me?
morning, sir. I'm audible.
Yeah, you are audible, sir. Can you hear me, sir?
Yes,
Dr. Paul
sir.
Raj?
Yes,
Okay.
yes, please.
Okay, perfect, sir.
Okay.
We'll just wait for 5 minutes more and we'll
Okay.
start. Okay. Thank you, sir. Uh, Hello. Good morning, everyone. Can you please confirm? Am I audible?
Yeah.
Okay.
Yes, sir.
Okay, great. So, a very warm good morning to everyone. I would like to welcome you all to day 2 of the seminar. A big thank you to all the participants who have joined us today as well. Your participation makes the 50th anniversary celebrations of ISPC all the more special. So, without wasting much time, let us kickstart today's proceedings. Metagenome sequencing has played a pivotal role in identification and characterization of microbial species from environmental samples. And that is what our first topic of the day is going to focus on. The first talk for the day will be presented by Dr. S. Paulraj. He's a senior scientist at ICAR CPCRI, and he would be talking about deciphering rhizosphere microbiome for sustainable crop production utilizing metagenomics approaches. So I would like to invite Dr. Paulraj to take over. Dr. Paulraj?
Yes. Good morning to all. I'm audible?
Yes, sir, you are perfectly audible, sir. You can start screen sharing as well, sir.
Good morning to all. I wish to deliver the lectures on deciphering rhizosphere microbiome for sustainable crop production utilizing the metagenomic approaches, because we are in the position to stay for the sustainability of the earth and also our agriculture stability of all the crop and their production potential. For the sustainable crop production, we should think of the soil, plant, and microbiome interaction. Soil is the major factors that influence the plant growth and yield. The plant factors, the plant genomic parameters that also have high impact on microbes also. The soil microflora that have high impact, high influence on the crop productivity and sustainability of the soil environment. When we think of this soil, plant, and microbe interaction, the rhizosphere region is the key niche environment compared with others niches like phyllosphere, spermosphere, and others niches. The soil is the major factors. Soil physiochemical properties play important role. Primarily, the parent material produced the soil matters that influence the clay silt composition, that influence the structure and texture of the soil in a soil. Then the organic matter content in the soil conditions and the crop residues, other materials we are added like some fertilizers, sand, silt, clay, gypsum for the reclamation purposes, that also have the high influence on the interaction, microbial interaction in the rhizosphere. The plant, if you think of the plant, the C3 and C4 plant have higher difference because the root exudates pattern varies between the species. C3 plants exudates most of the sugar components. C4 plants exudates most of the inorganic acids and other flavonoid compounds. The age of the cultivars, the type of cultivars, and the annual and perennial nature of the plant species that influence the root exudates pattern. The root exudate is predominantly dominated by most of the sugars like monosaccharide, disaccharides, and oligosaccharides, and protein, some amino acids, mostly a variety of amino acids. The organic acids are produced. These organic acids helps to establish the root to obtain the unavailable nutrient from the soil environment, and also to neutralize the toxic elements in the soil environment. Comparably, phenolic substances and other high molecular weight substances also produced by the produced into the by the plant has deposited as a rhizodeposit in the rhizosphere region that highly influence the rhizobiome of the particular plant or a particular crop species, and also the highly influence the core microbiome, which is highly influenced by the varietal parameters and other plant factors. Apart from this plant and soil environment, the microbe interaction in the soil environment also high influence on the rhizosphere impact that affect the relative abundance. Comparably, the other parameters like temporal and spatial dynamics also influence the rhizosphere effect on the plant system. The rhizosphere effect have several eco services. That is, the main factor is the life of the soil. That is the soil organic matter decomposition and mineralization process, and also the productivity of the crop and agriculture productivity that is linked with the availability of plant nutrients, which is by diazotrophic and mineral solubilization and multi-nutrient solubilization potential of the plant growth promoting rhizobacteria, and also the production of siderospore that suppress the pathogenic flora in the rhizospheric environment, and the antimicrobial substances produced by the bacteria are the PGPR substances that helps to overcome the disease effects, and the antioxidant substances produced by the bacteria, predominantly the rhizobacteria, that helps to overcome the effect of the stress, oxidative stress due to the plant metabolic reactions. Other things, apart from the beneficial activity, the plant diseases also covered by the microflora in the rhizosphere environment, like root wilt and rot diseases. Another important factor is systemic acute resistance and induced systemic resistance produced by the inoculating microflora or the beneficial microflora. Apart from, they also helps to alleviate the abiotic and biotic stress alleviation, specifically the ACC deaminase potential PGPR that helps to overcome the ethylene stress. These plant growth promoting and rhizospheric bacteria also have the higher impact on not only the production of plant growth promoting substances, but also the regulation of these substances in the plant systems. And these organisms in the rhizosphere and the root environment helps to bioremediation of toxic elements and the non-constituent materials in the rhizosphere environment, and also most of the photosynthete, up to nearly 30 to 40% of the photosynthete is diverted towards the rhizosphere environment. This environment, whatever the photosynthete that will helps to this microflora in the rhizosphere environment helps in the carbon sequestration also. My topic is to cover the metagenomic approaches. The metagenomics means the genome of the entire complete microbiota in the representing in the rhizosphere region by amplifying using the concerned region of bacteria or 18 or fungal. For bacteria, 16S hypervariable region interspace between the concerned region, that is nearly 9 hypervariable region is there. This region can be amplified using the specific primers. For fungal, the specific ITS primer used for this to develop the metagenomics data from the particular niche environment. These organisms, this metagenomic study help us to shift the individual microorganism bioinoculant technology to the consortia approach, which is a complementary and synergistic in nature, and also to develop a minimal microbiome strategy for the niche saturation to satisfy the particular ecological services in the agriculture production system, either plant production aspects or protection aspects. Normally, we practice mostly culture dependent isolation practices till now, because this technique have the major drawback that have the capacity up to 1% of the existing flora. But with the assistance of the metagenome approaches, we can identify and characterize the more than whatever the unculturable flora. When we are thinking of the cultural dependent technique, we should work out each niche's environment, each physiological potential of the each organism by the theoretical knowledge, and thereby we should design the media and give the chance to grow. This is have the different media we should evaluate. After that, we should subject for the different parameters like PGBR potential, other things. Then we should characterize. We should go for the DNA extraction. Then finally, we come for the identification of the organism. But using the microbiome approach, we can go for with the genomic DNA, total genomic DNA available in the rhizosphere environment, comprising of all the viable flora, rhizobacteria, or prokaryotic system in the rhizosphere region. We can extract the total DNA, and using the specific primer for the amplification of the specific region, we can go further to study the diversity, and also we can go for the specifically the particular activity using the specific primer for highlighting the specific activity of the particular microflora. With this, I want to highlight the single case study highlighting the areca nut yellow leaf disease management using the metagenomic approaches. The areca nut yellow leaf disease, the primary causal organism is phytoplasma. That is a biotic factor inducing the stress. Apart from these biotic factors, all other factors also have the positive effect on this stress, like the occurrence of nematode, wilt, and root rot pathogens, and the edaphic factors like the nutrient imbalances, typically the cation imbalance, because these areca nut plantation crops is in the acidic soil environment. So there is a high chance of imbalance of the cations like calcium, magnesium, and potassium. And also this soil acidic environment, the nutrient toxicity like aluminum, copper are very predominantly reported. This also have the impact on the stress on the plant system in areca nut palms. Apart from this areca nut leaf, areca nut plantation is in the hilly terrains or a slightly elevated terrain with receiving high rainfall, average of 3,000 to 4,000 mm per year. So, these yellow leaf diseases predominantly occurred specifically in the during the mid-monsoon seasons. Typically, the leaf yellowing, the very typical symptom compared with other micronutrient deficiencies and toxicities, the very unique difference. This due to the poor drainage, which is high rainfall condition, which saturate the macro and microphore of the soil and rhizosphere environment, that make the anaerobiosis system, that also induce the stress. Combinedly, all these factors induce the stress and leads to yellow leaf disease. This stress, based on the plant, the areca nut palm reacts to stabilize it by inducing the cytosol calcium level, because once the stress is there, to regulate the osmotic balance, they will absorb more calcium. Due to the excess calcium accumulation, that calcium accumulation leads to callose accumulation in the sieve tube elements, because the phytoplasma concentration is higher in the region of functional sieve tube elements of the root system of the areca nut palm. This makes, when the cytosol calcium level is increasing, that make calcium accumulations. The calcium accumulations, callose accumulation in the sieve tube elements, that infer the xylem transport, phloem transport of the photosynthase. These also this higher calcium level in the cytosols affect the calcium, magnesium, other cation imbalance. This calcium, magnesium are predominantly affected by this elevated calcium level. When the elevated calcium is there, because that when the elevated calcium is level, the plant system want in the where is the limitation of calcium in the soil environment, the plant system and metabolically produce the similar organic substances that mimics the calcium to alleviate the stress, that is the putrescine. The metabolically that is equivalent to the calcium level, so that will substitute the calcium in the cell cytosol system. This putrescine, that is polyamine, is a high energy consumption process. Thereby, the metabolic energy is highly diverted towards the synthesis of putrescine. High putrescine accumulation is reported due to phytoplasma infection in most of the crop species. This this disturbance in the metabolic activity leads poor carbon partitioning and photosynthetic accumulation in the source, that leaf, and poor translocation of the photosynthate through the phloem, that makes the leaves darker initially. Then more photosynthate accumulation in the root system or plant system makes the root system to exudate more anions, that more leads to leaching or oozing out of the root exudates, and finally disintegration, and finally the rotting of the entire root system. That is the typical symptom of the areca nut yellow leaf disease, because the root is predominantly damaged by this plant physiological interference. Then the another major factors is the ethylene accumulation, because the ethylene is the senescence hormone. When the plant system is there, when the plant system is under stress condition, initially that will produce the ethylene. When the stress is increased, other things, higher rate of ethylene production leads to transformation of all the green leaves into yellow in colors. Finally, the plant gets senescence and wilting in the very extreme condition. Because this is yellow leaf disease of areca nut is not a lethal disease compared with coconut yellow disease, but that is suppress the health condition of the farm. That will after 4 or 5 years that entire productivity came down. To overcome the things by including the rhizobacteria as a tool of management, because the rhizobacteria we have the higher potential of producing the ACC deaminase and also the multiple nutrient solubilization potential and growth hormone potential by utilizing the root exudates and other precursors like tryptophan from the plant system. These produced substances can be available to the plant system that stabilize the growth hormones level and also the nutrient imbalances. Because these things, the SAM is the precursor for ACC, aminocycloperin carboxylase, that is produced in the cytosol system. ACC synthase enzyme responsible for the production of ACC. That is essential for the metabolism reactions. When there is excess ACC is produced, that will be converted into 3 other forms like mannonyl ACC and glutamyl ACC and jasmonyl ACC. These mannonyl ACC is intermittent form, that is reversible immediately, but this jasmonyl ACC, that is once it converted into jasmonyl ACC, that accumulate in the root system, that leads to the decay of the roots. Thereby the entire root system get damaged, more ooze out came, and finally decomposed and wilted root system that affect the plant nutrient uptake further and intensify the disease incidence. These ACC can be converted into this. When the ACC is produced at the low level, then ACC oxidase helps to produce the ethylene. Ethylene is a growth hormone at the lower concentration. When the higher level of ACC is there, that stimulate when the iron and ascorbate in the phloem sieves, because the acidic environment, the iron accumulation is more, and growth cycles, the ascorbate production also there, that will help to induce more ethylene production, that leads to the yellowing intensity very higher during the monsoon season. And we can realize that due to monsoon season, the ethylene production, that cyanide is the subcompound that can be, we can visualize the fallen nuts which is in darken in color. This ethylene is the key factor that influence the stress during the monsoons. To overcome the, to develop the microbiome strategy for the management of yellow leaf disease, the microflora associated with the yellow leaf disease arecanut palm have been studied in our campus by using the metagenomic, that is the tag-based approaches of V3, V4 domain of 16S RNA hypervariable region, and also shotgun whole genome sequencing by using Illumina sequencing with the help of and the assemblage analysis with the Silva database. These results infer that there is a classical diversity index very high in the all the samples on the, because that is the organic matter rich environment, high nutrient rich also. So the diversity of the all, most of the genera of the is higher in all the things. When compare the healthy condition with the disease, the healthy condition have higher classical diversity index, because the healthy condition supports more root exudates and more root functionality, thereby they support the more diverse bacterial flora in the rhizosphere environment. When we compare the beta diversities, when we compare the two samples, that is a very significant OTUs are exist. But when we compare more number of groups in comparing groups, there is the significant OTUs is not, that infers that the population of the study is on sampling is limited. Even though with the what is the available data, we can come for some management strategies, because the relative abundance in the rhizosphere regions, predominant flora and the phyla is total nearly 10 predominant phyla. We can came for conclusion by the using the data. Among these, proteobacteria is more common and predominant bacteria when compared to other plant species also. And other predominant bacteria is the Bacteroides, bacteria, and acidobacteria and fusobacteria. Metabolic role, the heterotrophic fermentative lifestyle. So they are abundant in both rhizosphere and rhizosphere. Compare the healthy rhizosphere, the predominance of alpha proteobacteria and common proteobacteria and delta proteobacteria is at the phylum level, and also the phyla radiation bacteria like Paticibacteria and Planktomyces also very high abundant in the healthy rhizosphere soil samples. These Paticibacteria is the subsurface miniature bacteria that predominantly percolated from the higher terrain of the soil and moved and accumulated and enriched in the rhizosphere region. Another is Planktomyces. These Planktomyces are also the unique bacteria, but that is a very unique. That is no anamax group of Planktomyces exist in these rhizosphere regions of arecanut. Come this Vericomicrobidia, that insect associated group of bacteria, and cyanobacteria group, that photosynthetic nature of the bacteria also exist in the conditions. These, when we compare the family level using the data, predominantly the Xanthobacteriaceae, that is the alpha proteobacteria group is very predominant, and these predominant is most of the unculturable in nature, not much studied also. These xanthobacteria have higher potential of polyamine production. Then another is the nitrosomonadase, that is a key parameter in the key families involved in the nitrogen cycles. There is nitrification and denitrification process, and also that fermentative and spore producing microflora like Bacillus family is there, but the relative abundance is comparatively less in the healthy environment, but apparent healthy environment is there, but overall rhizosphere higher level is very much limited. When we think of the acidobacteria group, the acidobacteria subgroup 6, that is a very predominant in the rhizosphere environment, because these subgroup have the potential to grow in the low low cation exchange optimum neutral pH condition. These acidobacteria subgroup 2 under aluminum stress condition that will predominate, but when the healthy plants having the optimum cation exchange for sites, that aluminum stress is limited, so that abundance is not exist in the healthy environment conditions. Like subgroup 7 also similar to subgroup 6 characters. And the Paticibacteria group, that a Parcobacteria, that is key Paticibacteria, very high abundant in healthy and rhizospheric environment compared with the non-rhizospheric environment. And the Planctomyces group, different families like Gemataceae and Pyruliaceae, all the families Planctomyces are abundant in the healthy rhizosphere environment. This gives that this also, there is no Anamax bacteria. These also have the higher potential of putrescine type of polyamine production. This helps to support or the accumulation of the polyamine during the stress condition. These are similarly the nitrosomonadase is very high in diseased condition. This is the denitrification loss of most of the nitrogen. This group of bacteria also there. When we compare the chloroflexi, high abundance of KD4 phyla is there. It is having the hemoorganotrophic nature, so they are they multiply in the rhizosphere region utilizing the inorganic materials. When we compare the predominant genera, we first the Xanthobacteriaceae. Xanthobacteria is unclassified, and another genera is Pseudolabris, that is poorly explored till now. That is very predominant in the non-rhizosphere region also. When we compare also that rhizobials group, typically the Bradyrhizobium, because that is a non-leguminous plant. Even the occurrence of Bradyrhizobium in the healthy environment also very healthy rhizosphere notice. When we compare the nitrosomonadaceae, this is key parameters for the denitrification process and nitrification in the unnitrification cycles, that also abundant in the healthy diseased environment. Other heterotrophic bacteria like Pargolderia, beta proteobacteria, Acinetobacter, E. coli type also abundant in non-rhizosphere regions, also in healthy rhizosphere regions. So when we are the culturable isolation, these abundant bacteria only we are able to cultivate, because when we are using the heterotrophic culture media, these whatever the abundant in the soil environment also abundant in rhizosphere. So we are getting these type of flora only like Bacillus, Acinetobacter, and Pseudomonas and Burkholderia. When we think of the yellow leaf conditions, these bacteria, that is delta proteobacteria abundance, like Heliangium, Anaeroma oxybacter, and Geobacter, that abundance in the health diseased environment, because these are compared with this denitrification process, this further denitrification. without the Anamax enzymes or the nitrate reductase also, they use the iron substances as a electronic receptor. By the way of oxidizing the ion, they reduce by the way of reducing the ion in the rhizospheric environment, they oxidize the nitrogen into elemental nitrogen. That's why they leads to higher nitrogen loss in the rhizospheric environment in the diseased condition. Other bacteria like this Patysi bacteria, that is a Parcobacteria, other group of Patysi bacteria, higher abundance. These Patysi bacteria and Planktomyces are higher abundance in the rhizosphere, either healthy or disease condition, compared with the non-rhizosphere condition, because these organisms require very little amount of carbonaceous substances for their growth. They can survive in the limited nutrient condition also, but they have higher potential on the all the nutrient cycles, nitrogen, carbon, and other nutrient cycles, that also have the higher potential of the polyamine synthesis, that is a putrescine like substances that helps to mimic, helps to alleviate the stress, either biotic or abiotic conditions. When we work out the whole genome of the by using the Illumina, it infers that high abundance of heterostrophic rhodopseudomonas, high abundance of rhodopseudomonas that have the higher different pattern, that is hemo-organotrophic, autotrophic, phototrophic lifestyle. This environment, because this rhizospheric environment, most of the exudates, the hydrogen ion is released. That ion can be combined with the iron oxides and iron hydroxide transformation is existence. So whatever the hemo-organotroph or hemolithotrophic organism is predominant. But by culture dependent isolation, we missed the isolation of this type of Rhodopseudomonas bacteria in this process. The very high abundance of Anaeromoxybacter bacteria, that is indicated the denitrification process in the diseased environment compared with the healthy environment. Other things, the culture dependent isolation, we are getting the Burgholderia, Bacillus, Cupriavides, and other things. But these bacteria we are not isolated by the culture dependent technique, because we not know that the predominance of these bacteria. These metagenomic approaches, by this simple study, we came to know that we should study the different metabolic, study the ecological environment and their physiological nature. Thereby we should subject to the selective isolation. Then only the whatever the organisms we are getting for to develop the bioinoculant, that will work out. My studies, I missed the Rhodopseudomonas isolation. With the support of this data, I am trying to isolate this predominant Rhodopseudomonas isolation, because this Rhodopseudomonas is also reported as a very good PGPR. Another thing, these unculturable flora, some different bacteria like Acidobacteria and Patysi bacteria and Planktomyces, this is not thoroughly studied. If you have worked out for the isolation of culturable candidate of this particular species, that can be combined with this other cultural isolates, thereby we can develop the very sustainable bioinoculants or the microbiome to satisfy the particular niche. Because this YLD, yellow leaf disease niche, is highly influenced by the combination of all the biotic and abiotic stresses. So we should first thoroughly understand the physiology in the rhizosphere environment and the organism which is associated in the rhizosphere region, to support the to get the support of the rhizosphere through root exudates and the environmental factors for the survival. Then we can develop the predominant microbiome for the management of this root wilt disease. because we are these till now we have the diversity studies only to identify the particular type of organisms, that but the real things is should be on the function based diversity, so that we should study the different function in the rhizosphere environment influenced by the plant system and also by the microorganisms. These studies can be performed by RNA chip method or DNA microarrays, and also through the use of metatranscriptomics and qPCR and complementary DNA analysis supported with pyrosequencing. These are after combining this metatranscriptomics and metabolomic, that is the metabolites released and influenced the interaction, plant microbe interaction should be thoroughly combined, and then that will help us to develop the complementary consortia, that is the minimal microbiome, to alleviate this OLD stress. Again, identification of specific, because while grouping the samples, we are unable to get the unique microflora, because very the population dynamics of the microbial rhizosphere niche is very smaller in size, very small. When we work out it, so we should work out for the site specific nutrient management, site specific microbiome strategies, and also the different varietals of the some areca nut palms or varieties have, or wild types have some resistance. So we should develop the crop and varietal specific things strategy, and also we should work out other niche environment like endophytic nature of the microflora that also highly influence the plant system, that should be combined together, and finally we should come for the final microbiome for this management of areca nut OLD disease. This I wish to express my heartfelt thanks to the director of ICRCPCRA to execute the work, and also the previous director to initiate for the initiation of work, and also our head of division for Ravi Bhatt sir for mentoring, and other senior colleagues Rajesh and SV Ramesh and Hegde sir, and the soil scientist Priya associated in the project, and also express my heartfelt thanks to the Indian Society of Plantation Crops and Bionivid, the organizers of this webinar. Thank you.
Thank you, sir. Thank you for your talk. It was really interesting and informative. So, I would request everyone to put their questions in the chat box so that we can share them with the speaker and he could answer them. So, please drop in your questions. Ms. Tabia is conveying thanks to sir for his insightful lecture. Yeah. So we have Dr. Gina who is asking, can the microbiome be also applied to other plantation crops like coffee, which is grown in some places under Aracanid compared? I mean, she wants to know the comparative approach.
Normally, the core microbiome is highly influenced by the root exudates. The root exudate is governed by the plant genetic factors. So the rhizosphere microbiome and core microbiome of any individual crop should be highlighted while developing any microbiome strategies or inoculant technology. So that may work out, what, because they are sharing the common acidic environmental conditions and the common soil edaphic condition, but the survival based on the root exudates pattern in the individual crop. So that should be studied before developing the commercialization, but preferably go for the isolation in the rhizosphere region of particular crop and use it as a bioinoculant. That is commonly recommended.
Thank you, sir, for the answer. I hope Dr. Gina's query is addressed. And I have another question from Tabia. She is asking, I just want to know about what should be the sample size for the calculating the diversity indices for microbial diversity. What should be the sample size for calculating the diversity indices for microbial diversity?
Because the sample size means biological replicates, we should ensure the uniformity of the biological and technical replicate. Even if you work out the uniform higher sufficient biological and technical replicates, this study is performed when compare the two samples, that we are getting the significant operational taxonomic units between the samples. When we are grouping it together and we are combining the multiple group, we are unable to get operational taxonomic unique significant taxonomic units. That is really a failure of either statistical tools mismatch these studies or the real need of more number of samples, because we are working on the niche environment microflora, like that should be work out like the universal human population and studying taking a unique population the particular location and comparing the all the things, so that a difference will be there. That should be worked out to get the even the significant OTU using the multiple groups. Then only we can highlight that as a core microbiome for the specific crop universally. That is that much level sampling should be worked out. That is the particular unique location studies. So only we are unable to get the operational taxonomy significant taxonomic unit using the combining the different groups of the samples.
Thank you, sir. I hope Ms. Tabia's query is answered. We have another question from Amar Ganesh. He wants to know, for root module growth, how far this approach will be useful?
What repeat, ma'am, please?
For root module growth, how far this approach will be useful?
Because the root module, because that is based on the high this microbiome is highly influenced by the soil and plant system plant factors. This root system is highly governed by the plant factors when we are using different substrate for the growth. So the whatever the microflora, the core microflora and the particular root system, we can use it for to study the root modulation. Even because the root modulation is also governed by the balancing of different growth hormones. That also, while studying the things, the level of hormones should be worked out to get the desired modulations. Because if you higher level of IAA, that root elongation, that will stimulate root elongation, higher ethylene or ABA level, that will make more fibrous roots, more hairy root development. Like that, different modulation we can work out. By that manner, we can design or we can develop the different combination of bioinoculants to stabilize to modify the root system.
Thank you, sir. I hope Mr. Ganeshan could understand and get the answer. And now we have a question which is coming from YouTube. Sir, could you give us an idea about minimum number of raw reads required to proceed ahead with such type of analysis?
Sorry, I don't have that idea. Minimum number of how much required, because these ideas we have more than 44,000 in the 16S sequencing combinedly. I don't have the specific answer for this because I am not having much information on meta
Okay, sir. Thank you.
bioinformatics.
Thank you, sir. So one more question is coming. So I think we are done with the question and answer session. Thank you so much, sir, for the insightful talk. And I would like to invite Mr. Avinash Dhar. He's going to share a talk on an optimized NGS data analysis approach, the Genome Station experience. Mr. Avinash Dhar is working with Bionivid, and I will hand over the stage to him. Sir, can you stop the screen share option?
Okay.
Yeah. Thank you, sir. Avinash, please take over from here.
Thank you, Kranti. A very warm good morning to all of you. I hope I'm audible to everyone.
Yes, audible.
Okay, thank you. I hope my screen is visible to everyone.
Yes.
Okay. So a very warm good morning to all of you. My name is Avinash, and I've been working with Bionivid Technologies Private Limited for the past 4 years. And in today's session, I'll be discussing about an optimized NGS data analysis approach, the Genome Station experience. So we'll talk about the genomics revolution. How exactly did this all pick up? So in the year 2000, a press conference was held in White House to announce an extraordinary feat, which is the completion of the first draft of the human genome, which later, as we all know, got published in the year 2003. This was a landmark experiment in the history of mankind because a lot of scientists from 5 different countries, 15 different research institutes collaborated to decipher the mystery that was the human genome. And again, for the very first time in the history of mankind, we were able to read all the letters that constituted the entire human genome, 3.2 billion to be precise. And if we talk about the progress that has been made in the field of genomics since then, well, instead of 13 years that took us to sequence the first human genome, we have brought down the time to around say 3 days, and the cost of sequencing has come down from $3.2 billion to around $1,000 per genome. Now, this experiment kind of acted as a precursor to all the developments and advancements that took place in the field of genomics and sequencing technologies in general. And if we talk about the current day and age, this technology has become an integral part of all the modern research that is happening in the life science sector. Post this experiment, there was a flood of genomics data, not only humans or mammals to be precise. A lot of genomics based experiments began to be conducted in the field of plants and specifically crop and plantation crop research. So if we talk about the amount of data that this entire domain is generating, it is expected to surpass YouTube and Twitter as the single largest source of data generation in the world, and it is expected to cross around 40 exabytes of data. If we talk as layman's, our imagination as far as the data size is concerned runs into TBs, right? Here we are talking about something 3 times 4 times more bigger than that unit. We are talking about 40 exabytes of data. Similarly, it has been also estimated that by 2025 we are expected to sequence around 1 zeta base, which is around 1 trillion billion bases per year. So you can very well imagine the amount of data which is going to be available out there. And it has also been estimated that there would be more genomes sequenced than there are people on the planet. And you can very well imagine the amount of data, again I'm stressing on it, the amount of data that this particular process is going to generate in the coming time. Now if we talk about a standard sequencing approach, there are two main approaches that we apply whenever we are trying to plan a sequencing experiment. One is a de novo sequencing approach, which is characterized by the absence of a reference, and the second one is a resequencing approach, which is characterized by the presence of a well characterized reference genome. And this approach applies to whatever molecule you are working on, be it DNA or RNA. Now if we talk about various applications that are being worked upon in the field of genomics, you have whole genome sequencing, which covers both de novo as well as resequencing approaches. You have transcriptome sequencing, wherein you sequence the mRNA content of a particular species in order to get an idea of gene expression. You are talking about targeted resequencing, wherein you specifically target certain genes or regions which you sequence, right, in order to, most common example being the exome sequencing approach. Then you have genotyping by sequencing, which was very aptly explained by Dr. Awad in yesterday's session. Then you have chip sequencing, which deals with DNA protein interactions. You have metagenome sequencing, a lot of which was discussed by Dr. Paul Raj in his session. You have small RNA sequencing, and you have bisulfite sequencing, wherein you study DNA methylation patterns. So these are some of the most commonly utilized applications in the field of genomics as such. Now, if we talk about the tools that are available in the field of genomics and data analysis in general, because once you generate the data, obviously the data will not present findings automatically in front of you, right? You have to analyze it and make sure that that you are able to churn out something meaningful from it. So if we look at the stats between the year 2020 2021, the time when most of us were under lockdown, self-isolation, right? 325 new tools for various genomics data analysis based applications have been designed and published. These tools cover various applications, right from alignment, wherein you compare your data against a reference genome, to assembly, wherein you arrange your data to form something meaningful, which mostly is a genome. So tools ranging to all these applications have been designed, developed, and published. We are talking about new age alignment tools like Sibelia Z, Callisto, Salmon, to more sophisticated hybrid genome assembly tools like Wengen, WTD, BG2, and Shasta Toolkit. And if we talk about every application out there, the list of sophisticated and advanced tools that are coming up is ever increasing. So it obviously is the need of the hour for us to upgrade our analysis pipelines and keep them abreast with the latest that is out there in the research world, right? Because sticking to a fixed pipeline will no longer will start making sense. So you have a pipeline fixed right now, most probably by the next 2 years, using the same pipeline to get your analysis done won't make sense. There will be better, more sophisticated tools to give you more insight into the complex and dense data that is the genomics big data. Now, all said and done, the biggest advantage of all these tools that are coming up is most of them, almost 90% of them, are open source in nature. When I say open source, I mean these tools are available, and you can download them, you can utilize them, you can improve on them if you have the right skill set. Now, all said and done, we have talked about the brighter aspects, wherein we discussed about various applications that are there in the field of genomics, the amount of data that we are looking to generate, uh, and all these open source tools which are available to us to analyze the data that we are generating. But there are problems which most of us as biologists face on a daily basis. The biggest of them is, as of now, your DNA sequencer does not produce a complete genome, which your researchers, which we as biologists can read like a book. Rather, uh, it churns out data in the form of shreds, like shreds of newspapers without any organization of the fragments. You have to arrange them on your own, right? Again, the data is humongous as far as size is concerned, size and volume is concerned. So you are not expected to analyze it or work on it manually. You ought to have tools, and tools which are backed by the optimum hardware to analyze and interpret those data sets. In addition to this, again, the amount of data that we are looking to generate, both in terms of the output that we are input that we are providing to our tools, and the output that those tools are generating, we need to have state of the art storage devices or mechanisms which can accommodate that amount of data. We cannot simply depend on these classical sources of data storage which we have been used to, which we are accustomed to, most of the times, like pen drives, internal hard drives, or external hard drives, right? We ought to have sophisticated state of the art solutions available that can help us to store the data and retrieve it whenever required. And this is the harsh truth as far as the informatics part is concerned. Sequencing machines have improved and have started to appear in more and more laboratories in the country, but the same cannot be said about state of the art infrastructure for computational analysis and data storage. That is severely lacking in most of our premier research institutes as well. So there is a reason behind this skepticism. People, researchers, they are interested in procuring resources that can help them crunch their genomics big data into something meaningful that can add value to their research. But there are few reasons that make them a bit skeptical about taking the plunge and setting up a computational facility in their own local environment. The first and foremost is the inability to figure out optimal configuration for their establishment. uh, Most of the times we end up overspending or underspending, and the setup that we procure does not cater to our requirement to its full capacity. Again, most of the tools that we had discussed previously, the latest one or the previously available ones, all of them are, in fact 95% of them are command line based. And there's a general fear of the command line, the black screen and blinking cursor in the mind of us biologists, right? So we refrain from going into that direction just because we are not that comfortable with the Linux or the open source environment and the command line as such. The third and biggest challenge is who is going to put this facility into use, because the command line, the fear of the command line is so overwhelming that most of us, even if we procure a facility, even if we have a computational setup in our lab or in our establishment, we end up losing interest because the fear of command line is too overwhelming, right? we have commercially available options as well, but there are quite a few players in the market that provide us with various toolkits for genomics data analysis. But their biggest two of their biggest limitations is A, they are expensive, and B, they are rigid in terms of the pipelines that they accommodate. Expensive in what sense? You have yearly reoccurring license agreements. On top of it, these tools do not cover for your hardware requirement. So it becomes all the more difficult for us to procure a system, because you have to pay an exuberant amount to renew your license for that particular tool. On top of it, you have to invest separately in your computational facility as well. Okay. And rigidity, because you cannot add and modify the existing pipelines. Many times what happens is your analysis requires a non-standard approach, right? And for that non-standard approach to materialize, you need to mix and match your tools accordingly. That is unfortunately not the case with most of our commercially available toolkits for data analysis. So till now we have discussed about the problems that most of us face uh, when we look to take control of our data analysis and set up a facility in our establishment which we can use to do the same. Now let us talk about the solution. Welcome to Bionivid. We were established in the year 2011, and we are an ISO 9001-2015 certified company. If we talk about our various offerings, we have been actively involved in various research based activities in institutes with institutes of repute. We have in past and are still working with various DBT funded, CSR, ICR funded institutes, and we have been providing sequencing and analytics based services to them. Close to 80 publications are a testament to our hard work and expertise in the field of genomics and associated data analytics protocols. We also are into trainings and uh, education domain, wherein we conduct regular online as well as offline training programs for students, researchers, and scientists alike. During this lockdown, we have been able to train more than 3,000 students, and the audience was spread all across the globe. We had candidates coming in from countries like Costa Rica and Czech Republic, to name a few. We are also heavily into developing hardware and software based solutions for simplifying genomics data analysis and storage. That is what I'm going to talk about in the coming slides. That is what I'm going to stress upon in the coming slides. So we have developed products, softwares like SQIT, SQDB, and GStack. SQIT is a simplified analytics platform wherein you can perform your analysis on your own without actually having to take the trouble of typing the commands on the terminal. You have a GUI, a graphical user interface, wherein just like you fill your regular forms, you can upload your files, you can analyze them, and you can make sure that your data is processed. We have an interpretation platform in the form of SQDB, wherein you can upload your Excel sheets and you can derive all the meaningful pathway related informations from the same. We have a hybrid cloud storage platform by the name of GStack, which doubles up as a project management platform as well. You can store your data, track the proceedings of your project, and manage, share, collaborate data as and when required. Now, what all this comes bundled up in our state of the art, specialized, and optimized hardware solutions, which we have developed in collaboration with Dell India Private Limited, the Genome Stations. Now, as a solution, what do we propose here? we discussed about all the problems. So what do we propose here as a solution? We propose a proprietary Bionivid product developed in collaboration with Dell, which boasts of an optimized hardware for better performance. We have made certain optimizations in the hardware, and we have observed 20% enhanced performance while handling genomics data. We propose a machine that comes with a customized operating system. We like to call it as the GSOS, the Genome Station Operating System. This operating system comes pre-installed with all the tools and latest data analysis pipelines which are out there, which are published, which are well cited. We pre-install all those tools in the operating system so that your machine becomes a ready to use plug and play device. A graphical interface based analysis package, SQIT, which simplifies the data analysis for you altogether. Apart from SQIT, a system which is embedded with these simplified automated pipelines for NGS data analysis for various applications as such. And above all, access to a hybrid and dedicated genomics cloud storage platform, which is GStack. So we present before you, as I said in the previous slide, the Genome Station. We have desktop level to server level solutions for the same. Depending on your budget, depending on your analytics needs, depending on the sample load in your laboratory, we have systems suiting each and every need and pocket. So this is the Dell India and Bionivid collaboration white paper. It can be found on Dell India website as well, wherein this is a case study rather, wherein the Dell management has reported how these optimized solutions which we have co-developed have made life simple and more and made it more friendly for biologists who generally are not that much into command line based analysis, right? Now see, there is already a lot of competition in the market, and we, as I have already discussed, there are a lot of customized pipelines available out there. What makes the Bionivid product, the Genome Station, stand out from the rest? Well, first of all, we provide complete hardware and software support for your Genome Station. Okay, any problem in the hardware, any problem in the software, in your operating system, in the tools that you have installed, we take care of that. We provide complete cover to your machine. We make sure that your system comes with all the required data analytics packages. All those packages come pre-installed in your system. As I said before, all you have to do is connect it to a power source and start using the system. From raw data quality control to gene network to finding variants, right, to plotting circular genomes, everything that you have in mind, anything that you want to interpret from your data, you can do that using the power of your Genome Station. Also, updates released over a period of 3 years will be plugged into your Genome Station free of cost. Also, we have 9 years of experience in genomics data analysis and close to 80 publications, as I had discussed below. So we also provide free consultation to our esteemed Genome Station customers so that they can utilize their systems to the maximum. Obviously, our analytics team with vast experience in the field of genomics make sure that you utilize the Genome Station to the best of its capacity, and we make sure that you put it to use and don't just use it as a regular system. Use it as a specialized machine for genomics data analysis. We also have on-site online trainings included with the overall package. So we make sure that we train all of you to ensure that you know how this machine works and how to make the best use out of it. We also help in sourcing skilled manpower with experience of working on genomics big data and high performance computational systems as well. So all in all, you can see that this particular product works as an end solution to all your problems related to genomics and genomics big data analysis, right from support to making sure that you are utilizing this facility to the fullest. We have it all covered, so that gives an advantage to this product. Moreover, as I said before, we have the machine comes packed with various automated analytics pipelines, so you do not have to invest separately in a toolkit and then separately procure a system altogether, doubling up your expenses. You don't have to do that. A single machine comes bundled up with everything that you require to make your data analysis simplified. So this is SQIT, which I was talking about previously. So this is a small command. It might not seem small because it is running into 3 lines and it involves a lot and lots of characters that we more often than not end up despising, right? So it's a very big command. It is for performing raw data quality control of your genomics big data, quality control being the first step of any analysis. Now if you look at this command, it involves a lot of parameters which at present all of you might not be able to understand, because obviously some of you might be new to this. And imagine remembering this command, writing it, and then finding out that once you press enter, a very silly error comes up on your terminal screen. It would frustrate you to the core, right? Now, imagine such kind of a setup wherein the same tools require you just to fill this form, this simplified form. Not to type the entire command, but just to fill in these forms and click on submit. The moment you click on submit, your analysis is done and you get a customized reports bearing all the necessary information from your data. Definitely makes your life simpler. This is what SQIT does, right? From RNA sequencing data analysis to analyzing bacterial genomes to performing metagenomics based data analysis, this pipeline covers everything that you will need to make sure that you interpret your genomics big data to its fullest capacity. Again, we have several automated pipelines here in this SQIT, as I said, right from getting a list of your differentially expressed genes to various plots and figures that are associated with it. This is just for one publication, which is RNA-seq. We have similar sort of reports for which I'll be showing to you in the later stages of the presentation, that cover various other aspects as well. So right from heat maps to volcano plots, you can generate anything and everything using this particular package, right at simply by using your mouse, just by a click of your mouse, nothing else. So there's no need for you to install specific R packages which are heavily used for these plotting these figures. There's no need for that. All you have to do is just fill a form, click on submit, and boom, you are good to go. You have all these plots, all these data inputs available in front of you in your system. Again, what you get access to with your genome stations, it is obviously a subscription based platform which we call as GStack. Now the problem with current storage options that are available with us, for example Google Cloud, Google Drive, if I have to talk about, is that they charge you for every GB of data that you upload, right? You constantly have to keep a track of amount of data that you are uploading. Also when we talk about genomics data, there's no segregation here. For example, you have to manually segregate your data, create folders and segregate them into folders pertaining to your raw data, to your assembly files, to your differentially expressed genes, to various plots that you are going to plot using your tools, right? So we have designed this GStack platform in a way that it also doubles up as apart from being a dedicated genomics cloud wherein the structure of the entire cloud is designed in a way that you can very easily segregate your data on the basis of the nature, on the basis of its nature. You can share and collaborate with various users of gstack quite easily. Apart from that, you can keep a track of your project, the various stages that your project is in. You can very easily keep a track of it using gstack gstack's project tracking features as well. So this is what it roughly looks like. You can keep a track of total space that you have utilized. You can keep a track of various projects and the amount of data that you are utilizing. So not just sharing of data, you can also keep a track of your analysis and the overall profile of the cloud storage that is allocated to you on the basis of your subscription. So this also comes as part of your Genome Station experience. GStack, a dedicated platform for storing and retrieving and keeping a track of your genomics projects and your genomics big data. Now, if we talk about various Genome Stations installations that we have done, so in a span of around 5 years, we have been able to set up bioinformatics facilities and labs powered by Genome Station in various research institutes funded by several national level funding organizations. The count is in excess of 100 individual units, and these include smaller desktop level variants as well as big rack servers as well. These premier institutes include universities and research centers like JNU, ICGEB, DU North Campus, IBSD, Junagadh Agriculture University, RCGEB, etc., etc. So, and to add on to this, the concerned the users, the Genome Station users, they are not just restricted to a particular domain. Like it's not like they are only working. We have only supplied Genome Stations to people who are working on transcriptomics or animal systems or plant systems or a particular system as such. These the users of these Genome Stations are as diverse as they can get. You name a system which scientists are working on, and you will find a Genome Station set up in a similar sort of a facility. So yeah, right from RNA-seq data analytics to eukaryotic whole genome sequencing, scientists working, our clientele includes scientists working on different genomics-based applications altogether, making it an all-purpose machine that simplifies your data analysis task. Now, this is a small chart of various Genome Stations and the installations that we have done in the past. So we are slowly and steadily we are planning an expansion into the unventured territories as well, and by the end of this year you would see a lot more areas being covered in red. Now I like to give you a small demo of how exactly one such automated application, that is SQIT, works. If you look at the video here, this is the login screen. Okay, so you can register yourself. You can register yourself as well. I have this demo starts from the login process, but if you are a new user, you are required to register yourself first. So once you register yourself, you will be allocated a login ID and a password, which you have to enter here in the login screen. Once you log in, this is your home screen and the various applications which are available for your pursual.
Bionivid workstation.
You can start with raw data quality control. You can start with RNA-seq data analysis. You can work on RNA resequencing data analysis. You can work with metagenome 16S RNA data analysis, and there are several other applications as well, right? Now, if I take this further,
ஓவரால பர்சன்டேஜ் பார்க்காதீங்க. நீங்க வாங்க. இதை பார்ப்போம். வேற எதுவும் தட்டி விட முடிஞ்சா தட்டி விடுவோம். எடுக்க முடிஞ்சா எடுத்து விடுவோம்.
பிரிண்ட் எடுக்குமா?
பிரிண்ட் எடுக்க PDF.
Just a second, sorry. Yeah. So if I take this further, let us see how raw data quality control, the one which I had showed you previously, is performed using the SQIT workbench. So you click on raw data quality control, you select the type of data that you have. We have selected Illumina here. Select the type of data that you have. You enter your details, as in you enter the project details here. Basically your project name, your basic QC parameters. You select your output folder, give a name to it, right? And if you can spot here, the question marks in front of each parameter here give you an insight into what should be the input, what exactly does this parameter perform, what function does this parameter perform, and what exactly should be the input that you are trying to provide here. So you create an output folder, write your sample names, simple form filling, nothing else. You select your raw data, which is in FASTQ format, the forward and reverse read if it is paired end, just the forward read, just the single read if it is a single end sequencing approach. And you click on submit. So as you can see here, the analysis, if you go to the current instances window, you can keep a track of your project, the start time, the end time, what is the status, whether it is in process or it is complete. As is being shown here in the demo, once it is complete, you can click on the next step and you can directly go for report generation as well. Okay. Yeah, so this is the report generation protocol. Again, a small form you need to fill, bearing in all the details associated with the project. Once you fill that up, all you have to do is click on generate report and you will get a customized report specific to your data, bearing all the figures and information that you would need to interpret the overall quality profile of your data. So as we select our output folder here, and we click on generate report, so we get this report in PDF format that can be downloaded as well. So rather than you investing your time in learning Linux, getting to know how to type in the parameters, what does each parameter perform, all you have to do is fill up a form, click on submit, and you will have comments regarding the data and various other figures associated with the data right in front of you in a report format for your interpretation, right? Makes analysis quite simple, isn't it? I'll show you a demo report here as well. That would give you more understanding of the kind of interpretations and that you can generate using a genome station. So if you could excuse me for a second. Yeah, so If you can see the screen here, so this is a demo report which we generate. So you will have information regarding the samples here. You will have introduction regarding the pipeline that has been used, right? The various tools that were utilized in this entire workflow. You will have information regarding that. Then you'll have information about the steps, individual step by step, which you can refer from here. So data QC, you talk about the tool, you get to know about the various parameters that were used to process the data, the overall QC profile, that total number of reads, how many of them passed QC, what is the GC percentage, how many of reads were of high quality, how many of them were of low quality. Every information can be gathered from here. Then when you talk about alignment, this is a resequencing screen share is not happening. 1 second. Yeah. So yeah, so you get all information about your samples. You get insight into read normalization, as in if your sample has excess depth, you are normalizing it using various protocols. Then you will have information regarding the alignment tools that were used, what are the overall alignment stats, how your reads have aligned, how many of your reads have aligned against the reference, how many of them remained unaligned, how many of them are multi-mappers. All that information you can generate here. Then this is the graph for the same, and this is sample wise distribution in terms of a table in a tabular format. We also make sure that we provide you with overall remarks regarding your overall data. If at each step we provide you with our specialized remarks regarding the data, how good is the overall data quality, how well is the data aligning against the reference, all that this information we try to provide you with expert remarks on the basis of our experience. We have added that part also in the tool here. Then when it comes to quantification tools that were used, so this report deals with RNA resequencing data analysis. I'll show you one more report for bacterial genome sequencing data analysis as well. So to find out correlation between replicates, you have the principal component analysis here, which we have plotted. Then heat maps to check sample to sample correlation. When it comes to differential data analysis, we have plotted the same using volcano plots for you to get an idea of how many genes are upregulated, how many genes are downregulated. The same can be reflected through this table as well, and you can arrange your genes on the basis of an increasing or decreasing log to fold change value, right? So we give you remarks as well, what all genes in your sample are upregulated, how many of them are downregulated. Everything is covered here. Then a heat map for the same, we have plotted here the heat map also. So you get heat maps as well. Generally we struggle plotting all these extra uh, plots, right? Because most of them are R based. Somehow if we figure out how to analyze using these complex commands and pipelines, the next step is to interpret those results and depict them using various pictorial representations. So that is a big challenge most of the times. So this pipeline that we have pre-installed solves that issue as well. Mm, yeah. Again, when it comes to significant biology and gene enrichment, so various pathways, cellular components, various pathways in which your genes are involved, your various gene ontology, basically biological processes, molecular functions, cellular components, everything can be charted out in this single report, right? So for the same, you have for top 5 geoterms, you have these dot plots, enrichment maps, gene concept networks to show relations between various functionalities and the genes which are involved in regulating those functionalities. So you don't have to separately do all of this. All of this is done just by clicking on a submit button, and you get a complete end-to-end analysis for your sample, for your RNA-seq sample right here. We also provide you with methodology so that if you are including that in your publication, you don't have to struggle for that as well. We provide that that also in this report. We provide you a map of your output directory, as in where all the output files are generated, and a small reference of the publications, citing various tools involved here, and a small acknowledgement also. So this was all about the reports which we generate using this particular platform. Now let me give you a small demo of our cloud storage platform, which is GStack. So just give me a second. Yeah. So this is how the GStack interface looks like. You have to enter the login details here in the user ID and password section. Once you enter those details, you have to enter the password. Once you click on login, this is your interface that you are led to, and this interface will give you all insights regarding the various projects that you have loaded in your allocated space, the size that they have occupied, the percentage that they have used, the stage till which the analysis is complete, various applications, and the amount of data that each of that application is utilizing. Everything gets covered here. So this is your project management platform, wherein you get to keep an eye on what stage your project is into, how much data is each application occupying, so on and so forth, right? Also, see, so here we are talking about the various stages that are involved in the typical stages that are involved in your project, and what all have been completed, what all are going on. So you get an insight into that as well. Now, if this is all about the project management platform, now if I have to give you an insight into where your data is stored, well, this is how your data storage platform looks like. Data storage interface looks like. Segregated folders for each of your data sets, be it the raw data or the QC based files. You can upload one in 1_QC folder. Once you're done with assembly, you can upload it in 2_assembly folder. And depending on the kind of analysis that you perform, you can upload the same in each of your subfolders. Also, if you want to share your findings with someone, if you want to add collaborators as well, that can also be very easily done, because that is the true uh, power of any cloud based data storage platform, that you can share your data on the run with anyone whom you would like to do that with. Okay, so this was a quick demo of GStack. Now, as I said, we have installed genome stations in a lot of reputed labs and facilities. This is a small feedback that we got from Dr. Shailendra Goyal's lab. Uh, I would request all of you to just have a look at it. Was it audible?
Yes, you can.
Okay.
Genome Station. I'm from University of Delhi, Department of Botany, working as a research scholar. The broad area of research in our lab are molecular genetics, crop improvement, and genome organization. As you all know, this kind of work generates huge amount of NGS data, and for the analysis of same, we procured this small but very powerful instrument from Bionivid Technology Private Limited in 2020. array of tasks it performs are pretty very impressive, from small tasks like local blast to the big tasks like complete transcriptome assembly and DEZ discovery were very successfully performed by using this. Big genome data for comparative analysis were also achieved by this instrument. Also, as a user, I personally feel that this is a good addition to our lab, which has saved a lot of time because it had its pre-installed softwares and packages which are most commonly used in our research area.
So this was feedback from Dr. Shailendra Goyal's lab, and this is a desktop variant of our genome station. We have workstations as well as tower servers, what we call as tower servers also, and rack servers as well. So depending on the sample load, you can procure that. Now on as parting notes, I have with you a small framework for an ideal facility, that if you are looking to set up a dedicated genomics uh, data analytics facility, this is how it should pan out as. You should have at least 5 individual nodes so that the research groups associated with that particular lab can access those nodes. Preferably 64 GB RAM with Core i7 processors. Then you should have a centralized rack server which would be connected to all these nodes, so that if any of the groups is working on a heavy data analysis project, then instead of running it on their smaller nodes, they can designate it to the rack server. Now the configuration of that server can vary. You can have a minimum of uh, 256 GB RAM. If you're working on de novo projects, then it should be definitely higher, because if you have to process a de novo genome, then the amount of RAM required, the RAM that you have in your system becomes quite important. So if I give you a straightforward calculation, 1 GB of RAM is required to process 1 million reads. 1 GB of RAM is required to process 1 million reads. That is a straightforward calculation here. So whatever is the amount of reads that you are looking to generate, you can contemplate based on this stat. So for de novo experiments I'm talking about, so you'll require a heavy RAM setup when you are looking to analyze eukaryotic genomes. Even bacterial genomes also, if you are looking to assemble, you need to have a minimum 128 GB RAM setup. So this centralized rack server will be connected to all these nodes, so that, as I said, in future, if any of your research groups is looking to work on a heavy duty research genomics based project, then it can be designated to the servers. Then you can also have a network assisted storage, which is also known as NAS. It can act as a centralized storage facility wherein data from the server as well as all your individual nodes is stored, and that NAS can be backed up onto the cloud server, which is GStack, so that you can keep a track of your project, how well it is proceeding, and what are the stages that your analysis is into, and you can easily collaborate and access your data from anywhere. Also, what is recommended what is recommended here is a centralized power backup for the entire facility, because voltage fluctuations can damage the SMPS of your systems. Now once this is done, I'll give you a short what I'll call it as the ideal processor RAM combo for your systems. So if you are into resequencing 2 to 4 samples, a minimum 32 GB RAM, 4 cores, 8 threads. 4 to 8 samples if you are into resequencing, 64 GB RAM, 6 cores, 12 threads. 8 samples and beyond, minimum 128 GB RAM, 12 cores and 24 threads. Similarly, if you are looking to analyze de novo projects, then lower eukaryotes, 2 samples, 32 GB RAM. Up to 6 samples, I think 6 is also too much. Up to 4 samples you can analyze in 64 GB RAM. And if your sample load is 4 samples and beyond, then you ought to work or set up a facility which has a minimum of 128 GB RAM and beyond. Now this is all about it. So I would like to end my talk here. Thank you so much for your attention. And in case if you have any queries regarding the system, you can mail us at info@bionivid.com, or you can even call us at 9535619191. Thank you so much, guys.
Any questions?
Thank you, Avinash, for your talk. So we have few questions here. The first one's coming from Deepak Singla. He's asking, can this toolkit work on Windows as well?
Most of the tools that are available for genomics data analysis, they are uh, Linux based. So and as of now, as an independent entity, our toolkit is not available. It comes bundled along with our GSOS, that is again shipped along with the genome stations. So as of now, it's not available for Windows, because most of those well cited tools that we have incorporated in these automated pipelines, they are Linux based. So yeah.
I hope Deepak's answer was addressed. Now we have Dr. A.R. Sherpa. His queries: is the genome station comes with all the softwares, toolkits, or we need to buy separately?
So, sir, what we make sure whenever we are, discussing uh, the genome station setup with interested clients is that we make sure we try to understand, as I said, the sample load that they are looking to analyze on this machine and the applications that they are working on. So based on that, we make sure that the automated pipelines are pre-installed in these systems pertaining to their applications. You don't have to pay anything extra for that. Even for that matter, as support, we, in case if you come across a new tool that you want to be installed in your system, we can do that remotely as well. It might not be incorporated into the automated pipelines, but if you want to install it separately, that can be very easily done. As far as the automated pipelines are concerned, I would repeat my answer that I had given previously. We make sure that first we understand the client requirement, and based on that, install all those automated pipelines that can help in making the client's analysis related efforts quite low and give him or her the maximum results. You don't have to buy them separately. You just have to pay for the genome station as such. It comes bundled in with all the automated pipelines that are for your project, that are required for the type of analysis that you do. No separate payment.
Thank you, Avinash. The next question is coming from Dr. Sandeep, ICAS CPCRI. Whether do this application use automatically any kind of reference database, irrespective of organism working with and generate results? How much the generated results are much reliable to make conclusion?
Okay, so we make sure that the user is granted maximum flexibility here. So any database that you are looking to add, you can download it and add in your folder. Okay, basically when I had shown you how to create a login in the system, right? So each login generates a user's individual folder, so you can download all the well cited databases there. Also, the results are 100% reliable because all the tools that have been incorporated in generating this pipeline are well cited and have multiple publications to their credit. And I can go up to an extent and claim that most of them are being used by us in our day-to-day analysis work as well. So you can pretty much rely and have faith in the results that you are getting through these pipelines.
Thank you, Avinash. We have few questions coming from YouTube as well. So here's a question from Krishnendu, and he's asking, "Sir, can you please name a few open source bioinformatics tools for transcriptomics data analysis?"
Yes, so if you are if you're if you are looking to work on de novo data analysis, then for assembly you have Trinity. If you are looking to work on resequencing data analysis, you have uh, for alignment you have Top Hat, you have HiSat2. Then for assembly you have Cufflinks. um, then there is Star Aligner. Even in de novo you have tools, a pipeline by the name of Velvet OSS that can also be used for assembling transcriptomes. Then for differential expression you have DSig2, EdgeR, CuffDiff. And for significant biology analysis you have various online repositories, like even you have a tool called Blast2Geo. Then you have online repositories like David, you have Uniprot KB that can be used for significant biology analysis as well. So yeah, these were some of the tools that you can use for transcriptome data analysis.
So he has another question. Are the tools the same one? Actually, we had the same one. So he wants to know, can we use these tools on Windows system or Linux system is must?
Most most of the tools that we utilize are meant for Linux based systems. You have Windows variants for them available as well, but the installation is a bit more complex. In fact, installation of these tools in any platform as such is a bit more complex. So yeah, preferably you should have a Linux environment to run these tools. Not all of them, some of them have Windows based executables available. You can utilize those tools on Windows. Like if I'm not wrong, Trinity has a Windows executable available, so you can utilize some of them there. I can see one question is from Mr. Deepak. Yeah, Galaxy, sir, as you said, Galaxy has inbuilt tools with GUI interface, but what we provide extra here is an automated report which will cover all the facets of your data analysis requirements. So if I talk about an RNA sequencing project, right from raw data QC to generating various plots like volcano plot or heat maps, you can do everything right from your just by the click of the mouse. Plus all of them gets incorporated into an automated report that you can utilize further to interpret your data. So yeah, that is something which is missing in Galaxy just I'll provide it. I can provide the link once the seminar is over. I can do that. I can help you out with that, sir. Once the seminar is over, I can share it over mail. Kindly drop in your mail ID. I'll do that. So purchasing this system from us gives you that advantage. Also, moreover, any latest tool that comes up, we install it for you. Updates are always there. We provide you, as I said, the consultation also. If someone who is unaware of how to analyze his or her data, we can guide them through. Obviously, we of course we won't be performing the analysis for you. That is a separate service altogether, but yeah, we'll make sure that you at least get a basic idea of how to perform the analysis. Plus there are trainings involved as well. Every genome station comes with an online offline training depending on the agreement that has been reached upon both the parties. So we train you on how to work on various pipelines. So yeah, that is an advantage that you get. Plus you don't have to separately here invest on when I talk about commercially available pipelines, you don't have to separately invest on pipeline and separately invest on the hardware. You get a combo of both. So yeah, support well characterized reports and support well characterized reports and ease of access for data analysis. That is what we promise here. Sure, ma'am. Dr. Deepa, you can call me anytime. I will drop my contact here. That is the company contact number. You can reach out to us anytime. In case if you need my number, it is 968215407 and my mail ID is avinash@bionivid.com. Any more questions?
There's one more question coming from YouTube. Can you arrange program how to do sequence using these tools?
We have training programs. As I said, during the lockdown we had conducted a lot of them. So our education team will keep you posted. Kindly drop your email ID in the YouTube chat section. I'll ask the concerned team to make a note of it, and they'll get in touch with you with regards to various training programs that we have to offer on these pipelines. Any more questions?
Thank you, Avinash, for the insightful lecture and introducing everyone to Genome Station. Now, I would like to invite Dr. T.
Okay, thank you so much, guys. Thank you for listening to the session patiently. I'll end the session here. Also, I would like to take this opportunity to introduce our next speaker. Just to give you a briefing about her session. Not only humans, we can say advancements in next-gen sequencing-based methods have revolutionized research in plants as well. Identification of SNP markers, understanding plant stress relationship along with gene expression are few such advancements that we have made in this particular sector. So to throw more light on various on such advancements, we have with us Dr. T.E. Shija. She's principal scientist at ICAR IISR Kochi. So I would request Dr. Shija to take the center stage and start with her talk. Over to you, Dr. Shija.
Hello, excuse
Yes,
me.
yes, ma'am.
Can you see my slides?
We can see a Word document here.
Oh no. Can you see my slides now?
No, ma'am. We are seeing just the Word document here. Abhi, the screen sharing has stopped. Yes, yes, ma'am. Now I think we can. You have a gray color background to your slides, right?
Yeah, yeah.
Yes, yes, yes. We can
Is
see
it
that.
okay now?
Yes, perfectly fine, ma'am.
Yeah, okay, thank you.
Thank you, ma'am.
So, a very good morning to all. So, first of all, I would like to thank the organizers of ISPC and Bionivid, as well as my director, Dr. Santosh Jayapan, for nominating me for this webinar. I'm working as principal scientist here in the Division of Crop Improvement and Biotechnology at ICR Indian Institute of Spices Research. So the topic for today is recent trends in application of next generation sequencing for improvement of spices. So actually, next generation sequencing was discussed in many of the talks during this webinar. And it encompasses a number of different sequencing technologies like Illumina, Roche 454, Iron Torrent, and solid sequencing, with a common feature that it allows you to sequence DNA and RNA much more quickly and cheaply than the Sanger sequencing. So this new massively parallel sequencing technologies, which gives you about millions or billions of reads, it's actually revolutionizing the molecular biology by providing insight into the genome, the transcriptome, the epigenome, actually on an unprecedented scale. So next generation sequencing produces short DNA reads, much amenable to fast analysis of the genomes. So coming to the most important application of NGS, it is definitely the RNA-seq, which is the best tool for the study of a transcriptome. So now you will be asking what a transcriptome. You must be already knowing what a transcriptome is. Transcriptome is nothing but the measure of the cellular status that gives you an idea of the nucleotides, amino acids, fatty acids, cofactors, etc. In fact, it is a collection of RNA transcribed in a cell at a particular point of time. It's composed of mRNAs or coding RNAs. In fact, the transcriptome sequencing or analysis of the transcriptome will promote understanding of the gene expression under different conditions and allow gene discovery, transcription patterns, so that you can understand more about the cell function and the metabolic mechanism. And it will also help you to compare the expression levels between organisms, between conditions, etc.
Sorry to interrupt, ma'am. Ma'am, could you please start slide could you please change it to presentation mode? Like,
Okay.
yeah, perfectly fine, ma'am. Thank you.
Okay. So coming to the spices, in fact, Coming to spices, you must be already knowing that spices are used since ancient times, like as preservatives, as colorants, flavor enhancers, and as medicines. And molecular marker identification is very much essential for improvement of traits as well as for germplasm characterization, for establishing varietal identity in spices and understanding genetic relationships. Because in spices, as you know, we have very huge germplasm repositories housing more than 2,000 accessions in a particular spice. So it is it becomes very cumbersome to maintain this huge germplasm. So it is needed that you identify core collections to cut down the size so that you reduce on the expenditure involved in maintaining this huge germplasm. You don't maintain the duplicates, in fact. And also varietal identity is very important. Since our institute, we are releasing elite varieties. We not want adulteration or spurious mixing up of varieties. So for all these reasons, molecular markers are very important. And definitely, needless to say, genetic relationship study is important from a point of view where you want to identify the phylogenetic relationships and from the breeding point of view. And these type of studies are very much prevalent in spices. And next important thing is the secondary metabolites in spices. Actually, spices are specialized specialized type of plants where you have a lot of secondary metabolites belonging to the classes for terpenes, alkaloids, phenolics, and all these contributed to the unique qualities in spices. So needless to say, the biosynthesis of these secondary metabolites and the factors that affect the biosynthesis are also gaining importance. But however, we have several limitations in the conventional methods, especially for unraveling these biosynthetic pathways involved behind the synthesis of the secondary metabolites. And I think it is at this point that techniques like NGS is very, very important, and it's very, very useful and handy. And needless to say, the transcriptomes and whole genome sequencing are the most important genomic strategies now currently practiced worldwide for identifying, not only for identifying markers, but also for unraveling the biosynthetic genes and the regulatory mechanism behind them. Actually, today in this seminar, I'll be briefing you. I won't be touching on the NGS part of it because already everything has been handled by Bionivid. So there is no need of repeating the same. But I'll be briefing on how I have utilized this NGS, how I have made use of this marvelous method for gaining genomic information from spices, for mining of markers, for genetic characterization and varietal identity, for evaluating the nutrient status in spices, for identifying trait-linked markers by association mapping, then characterization of certain important biosynthetic pathways, like identification of the structural and regulatory genes involved in these pathways, and also certain other regulatory molecules that are involved in regulation of these secondary metabolites in mainly I'll be dealing with two crops, that is turmeric and black pepper. Definitely there is a time limitation, so I will be actually my best. I will try to give you an overview of things, and for any detailed information, I request you to contact me directly. So this is actually what you're seeing on the left side is a previous slide. What you're seeing on the left side, it's the turmeric plant. Many of you might not have seen it. This is a turmeric plant, and this is the rhizome. And this is the edible useful part, which has lot of metabolites and medicinal properties. And this is the turmeric powder, which you can find in your kitchen. So coming to the turmeric, turmeric actually otherwise known as curcuma longa, it belongs to the genus curcuma and has a diverse uses in fields such as religion, traditional folk, modern medicines, aromatherapy, cosmetics, dye, floriculture, food industry, you name it. And about more than 80 species are reported in this genus. And turmeric is the most important member of this genus. But there are some problems associated with this crop, like low pollen fertility, and it is vegetative propagated. So there is very little scope for variation in this crop. And mainly it is propagated through clonal selection. And so far we have only 2 open pollinated progenies of turmeric in the world. And IS are about ISA germplasm houses about more than 2,000 accessions, which is actually a repository for the source of variations in agronomic traits as well as other quality traits. And definitely there is a confusion existing in the taxonomic revision of this species. We have to do the taxonomic revision is not done because we have a lot of confusion regarding occurrence of synonyms in this species. So just I wanted to show you some pictures of these important members of this family. And this is black turmeric, you know, it's very, very costly. And this is Curcuma aromatica, which is known as the Kasturi turmeric. And this is mango ginger. They have varied applications. Similarly, in case of black pepper also, you know, black pepper is most important spice in global trade. And India is the major producer, consumer, and exporter of this commodity. It originated from the Western Ghats, and in Western Ghats, we harbor the maximum genetic diversity of this crop. However, we have very little genomic resource. Definitely, we have the whole genome sequence available in black pepper now. That is a very good thing. And it is, however, a lot of research has to be done based on this genomic resource. And we need to identify markers essential for QTLs and genetic enhancement and crop improvement is yet to be done in black pepper. I definitely feel we have to take up more, you know, more NGS based work on this crop to actually fully utilize the whole genome data that is available. So in both this crop, the conventional taxonomic tools have been proved to be insufficient and challenging. And definitely there is no question that we need molecular tools. And whatever resources available in the public domain, whether it is EST or SSR or SNP, we have very limited information as of now. And there is an urgent need to develop these genomic resources in order to, you know, address all those issues, including the latest issue of biopiracy, like where people, other countries are stealing our germplasm. So this is one impending issue, IPR related issue, which also need to be addressed. So that's where we need such revolutionary technologies to address such problems. So that is how, you know, we had, actually in my lab, I had 3 projects, 2 funded by DBT and another by ICAR, wherein I was able to take up a little bit of my work on NGS. Just we have touched the iceberg, still much more has to be done. And we have a running scheme on cabin till 2025. The scheme ends only in 2025, wherein we are dealing only black pepper. So through these projects, I have been, in the last few years, I've been carrying out my work. And today, that is what I am going to present before you. And let us take up, you know, points one by one, applications of NGS one by one. And in fact, we do not have much information on this crop. NGS, as much information of NGS-based information in these crops. So coming to the first application, that is NGS for SSR discovery by RNA-seq. You know, in the normal way, any lab starts with basic work. You know, similarly, I too started my work by discovery of SSR markers from RNA-seq, mainly for, you know, for marker identification, for varietal identification. But definitely I had in the back of my mind, you know, one fine day I wanted to work on this biosynthetic pathway, because curcumin is supposed to be the most important compound. If any person working in turmeric, they should concentrate, at least put some energy, some work should be done on curcumin biosynthesis. That's what I felt, because it's the most important compound in turmeric. So with this idea, I had actually went in for Illumina HiSeq 2000, involving 4-month-old turmeric rhizomes from a high curcumin variety. Note that it is high curcumin. The curcumin content is sometimes goes up to 9% when grown in ideal conditions. And I compared it with Curcuma aromatica, which is 0 curcumin. Okay, so I just wanted to see the differential expression regarding the pathogens. That was one of main objective. Then I also wanted to look in for SSRs. So this is actually the summary of the transcriptome data what we have generated. Total RNA was extracted from the rhizomes, 4 to 5 month old rhizomes, because that is a stage at which the curcumin is maximum in the rhizome. So with a single transcriptome data, I wanted to not only isolate SSRs but also do my biosynthetic pathway work. So this is actually the summary of the transcriptome, length distribution, gene ontology search and everything. So ultimately, we could identify about 7,000 SSRs based on these transcriptomes, and only very few SSR, that is 4 SSRs, I could identify as related to the pathway genes, biosynthetic pathway genes. So coming to the SSR development, we had also some ESG sequences deposited by the Arizona University. So we had developed some EST-based SSRs from this data also. And we developed also the genomic SSR probes from this data. And this is how we had done the genomic SSR development from enriched libraries, following restriction, digestion, linear ligation. And this was a Dynavid enriched DNA protocol. And by this method, we could isolate several SSR markers. And these are the details of these SSR markers. And we could distinguish our varieties using these SSR markers. And not only we could distinguish our varieties, but also we found that these markers had cross-species amplification potential. They could be used not only in turmeric, but also in ginger and cardamom. And we further refined our resolution, started using this microchip electrophoresis system from CPCRI. And this technique, in fact, it helped us to identify most of our very closely resembling released varieties. And this research I've already published, some 4 or 5 publications have come out of this research. The next application is the DD-RAT sequencing and association mapping for quality traits. So actually, when you think about association analysis using SNP data sets, this actually helps us to localize the genes for various agromorphological traits and identify genome regions for mapping of traits with SNP markers. Marker-assisted selection is nothing but transfer phalings between gene pools, and it is based on closely and tightly linked molecular markers with the trait of interest. In fact, in association mapping, you do not need any molecular mapping. Prior information is not needed for establishing marker trait associations. So association analysis, you get that some of these SNPs, normally what you look for is the SNPs associated with certain markers and traits. So these SNPs are found to be, these marker trait associations are found to co-segregate. They are found to co-segregate. Based on this co-segregation at different locations in the genome, across diverse set of mapping population, you are ultimately able to establish this marker trait association. So here what we have done is we have chosen the NGS-based DD-RAT sequencing strategy for a genome-wide SNP-based association study for identifying useful agronomic traits, and also we wanted to track Kurkumin biosynthetic pathogens. And that's how we, this is a strategy what we have followed. It was based on linkage disequilibrium, and we had tried to establish correlation between genotypes and phenotypes in a collection of a germplasm of 92 curcuma long accessions, varying in curcumin content. Yeah, you have to note this point that the curcumin content varied from 0.5 to 9%. So whenever we found that LD is 1, we found that these two laws were closely related. And SNPs, we found that they are highly suitable for this kind of an analysis because of their biallelic nature, the codominant nature, and their amenability to high throughput sequencing. So we could generate a large number of SNPs from this project. And this is a list of accessions that we had screened. And we had scored the agronomic traits based on the DUX guidelines, and we also scored for curcumin content, oleoresin content, and oil content. I think major focus was on curcumin content, and the analysis was done following the TASL software we have used, TASL program, and then this is actually the summary of the work. We could generate about 16.6 GP data from this analysis, and about 2,000 SNPs we were able to successfully associated with agronomic traits. But interestingly, we found that some of these SNPs were associated with multiple traits, which indicates that either the clustering of genes for multiple traits, or even that the same set of genes at a particular genomic region, they regulate multiple traits. So suppose you see this particular SNP, it is this single contig is associated with traits like shoots and oleoresin. And we also, another interesting thing was that we found that multiple SNPs in a contig were associated with a particular trait. And we found that curcumin content and oleoresin, we could locate more than one SNP in a single contig. And for curcumin content, we could locate about 69 SNPs associated with these traits. And these are the top 10 SNPs associated with curcumin content. And now we are working on these contigs for further validation. Some unique SNPs, as an offshoot, we could also identify some unique SNPs which we could use for, characterization of to establish the identity of our released varieties. That is an extra added advantage from this project. So now let me go to the, I am slightly going fast because there are so many slides I wanted to show you. So in case you have any doubts, you can ask me later. So now coming to third and the most important application of this NGS, that is unraveling of biosynthetic pathways. Again, coming to turmeric, as I've already told you, the active ingredient in turmeric is curcumin or curcuminoids. And you already know that turmeric is known for diseases like Alzheimer's, immunity disorders, respiratory problems. And even we have come across reports in turmeric that it can be used to treat, some complications related to AIDS. So curcuminoid, actually it's a mixture of curcumin, dimethoxycurcumin and bis-dimethoxycurcumin, and it is collectively known as curcuminoid. And curcumin and its biosynthesis has become the major focus of attention internationally at present. And we have at ISR about 5 turmeric varieties like Pratibha, Prabha, Alapi Supreme, Kedaram, and Pragati. These are all high curcumin accessions where curcumin content ranges from 5 to 6.5%. And we have released these varieties for large-scale commercial cultivation. Actually, in our germplasm, we have observed that this curcumin content is varying from 0.5, as I told you, 0.5 to 9%. So we also observed that some interesting facts about curcumin is that curcumin content varies from between accessions and also from place to place, and it is subject to agroclimatic variations. It's very sensitive with G-to-V interaction. For example, if you take a turmeric variety, say for example, like Lacadon turmeric, which grows very well in Northeast, when you take Lacadon turmeric from Northeast to Calicut, the curcumin content drops from, say this is approximately 9% to 5 or 6% when grown in Calicut or in Kozhikode condition. Same thing when Pratibha is taken to Coimbatore, curcumin content drops from 5 to 6% to 1.5%. So this we found very interesting. So, and also on one side, we have an increasing demand from the industry side to enhance or stabilize the curcumin content or the extractable curcumin from turmeric. So that's where we felt that understanding the genes involved in the curcuminoid biosynthesis and its regulation is essential to manipulate this curcumin content in turmeric. So along with that, it is also required to study the regulatory molecules like transcription factors and microRNAs, which are known to promote or repress the biosynthesis by binding to DNA regions. This is actually the biosynthetic pathway for curcumin biosynthesis. And few of these key genes are already known. And see, you can see the turmeric rhizomes here. You can see the variation in color. See, these light or pale yellow colored accessions are literally devoid of curcumin. So once the curcumin content increases, the color changes from pale yellow to dark orangish color. So we actually employed our NGS-based RNA-seq analysis involving a high curcumin Pratibha and a zero curcumin Curcuma aromatica, and we have utilized 4-month-old rhizomes, and we in fact search for all the biosynthetic pathway genes, and we surprisingly we found that even in the zero-curcumin Curcuma aromatica, you can find all the machinery for biosynthesis. So then it was really it was interesting because there was something that is preventing the biosynthesis in Curcuma aromatica. There was something that was regulating the production of curcumin in aromatica. So then we went behind this, this fact, and then we started researching more. Then actually the transcriptome we further analyzed for some regulatory molecules, and surprisingly we found that there was a plethora of transcription factors which are known regulators of the pathway, and especially we could locate transcription factors like MIBS, BHLH, WRKY, which are known to regulate the phenylpropionide pathway in plants. Curcumin is a phenylpropionide derivative, so we suspected that maybe this differential expression in the transcription factors could be one reason behind the regulation of curcumin biosynthesis in the related species, that is Curcuma aromatica. So you can see the relative gene expression. See how these biosynthetic pathway genes are showing an upregulation in comparison with a zero-curcumin variety accession. And again, we verified the expression in different experimental backgrounds. First we took the different developmental stages, that is 60 days after planting, then 120 days after planting, 180 days after planting. We found that at the optimum curcumin biosynthesis, that is after 120 days, we have all these pathway genes upregulated in a high curcumin variety. And expression levels, we found that they dropped after 180 days of planting. There is a differential regulation. And we examined the relative gene expression in different environmental conditions, that is under Kozhikode field conditions, in Coimbatore field conditions, and also under greenhouse conditions. We found that the rainfed conditions in Kozhikode was an optimum, very much optimum for expression of these pathway genes, and we also correlated it with the metabolic content, that is curcumin, and we see the highest amount of curcumin, 6.52. It correlates well with the upregulated status of all these biosynthetic pathway genes. So you can see we did a Pearson coefficient, and this is the Pearson coefficient. And we found that all these genes were upregulated and they correlated well with the metabolite content. Actually, all these secondary metabolites are synthesized by multi-step reactions, and product of one reaction becomes the substrate for the other. So hence, the activity of each enzyme will depend on the amount of substrate produced in the previous reaction. So we found that all these genes are co-expressed, and these highly co-expressed genes are more likely to be correlated also, and they display a very good connectivity with them, and they influence the regulatory process in such a way that a strong co-expression relationship between a gene and a metabolite is actually the key to select a gene for metabolic engineering. So that is how we have established the correlation. And we have shortlisted, among all the genes we chose from the transcriptome, we have shortlisted them as correlated with the curcumin content, and those which are not correlated with the curcumin content. And finally, we realized that it is the polyketate synthesis. You can see here the polyketate synthesis with the highest R value, which are actually showing the greatest correlation with the biosynthetic pathway. So we went in further detail or further detailed analysis of the polyketate synthesis. So polyketate synthesis involved in this biosynthetic pathway. So then again we realized whatever transcriptome we generated was not sufficient, because it was between two different species. So we wanted to minimize the background noise. So we went in for a fresh transcriptome. We did a stress transcriptome with the single variety, that is single high curcumin variety, Curcuma longa, that is IASR Pratibha, which is a high curcumin variety, and we subjected it to nutrient stress, and we found that the nutrient stress will actually reduce the curcumin content from 6.5 to approximately 1.5%. So this was the exact background that we wanted to examine these polyketide synthesis. And we found that 12 unigenes were upregulated in the non-stressed curcuma longa compared to the stressed curcuma longa. And we also identified, we shortlisted all these polyketide synthase, diketide CoA synthase, and curcumin synthase 1, 2, and 3, and a novel polyketide synthase, which we named as curcuma longa polyketide synthase 11. And we found that all these key genes were upregulated under different, we actually planned experiments, field experiments involving different conditions to ensure that these genes were correlated in spite of the situations the experiment was undertaken. So if you see here, we evaluated them under organic, integrated, and chemical management practices. Then we evaluated them under different light regimes. We evaluated under nano zinc supplemented soils. Under all these conditions, we found that all these genes correlated well with the curcumin. So this is one indication that they are actually involved in the pathway for curcumin biosynthesis. So this is our stress transcriptome. You can see how we have shortlisted the genes. We even went one step ahead. We went cloning of the full-length genes. We wanted to see whether there was any sequence difference between the coding regions of the genes in high and low curcumin accessions. Surprisingly, we found that within the accessions, the coding regions did not change much. It was the same. In fact, it was the same. There was 100% similarity. So again, this fact again points to the fact that there are some regulatory molecules involved in regulating this biosynthetic pathway. So this is how we have cloned the full-length cDNAs of genes. And another interesting thing what we noticed is that each enzyme of this pathway is found to exist as isoforms with very minute differences in their gene sequences. And it is very important to identify the candidate isoform which actually is involved in the pathway. Not all isoforms are involved. So if you take if you go for public domain sequences, and if you take conserved sequences, and if you design primer based on these conserved sequences, and then if you start evaluation, you are definitely going to go wrong because you are not actually seeing the expression of the candidate gene. So this is how we have cloned. We have did a vast analysis, and then we found that it is indeed it's a polyketide synthase belonging, because it clustered with the polyketide synthesis of Musa acuminata, because we do not have earlier deposited sequences from our species. How we have amplified and how we have evaluated, I'll just skip through. Actually, we have followed a smarter PCR technique for full-length gene cloning. Now, this is slightly important because this gene is, that is a CLPK11, is a novel gene, not yet reported earlier. This gene, we found that we have now taken this gene as a bait gene or a marker gene to evaluate this biosynthetic pathway. We went in for a cloning of this full-length gene, and then we found that the phylogenetic analysis of this gene indicated that it clustered along with the other polyketide synthases, and it was a non-chalcone synthase gene. We even studied the amino acid residues. We studied the secondary structure of this gene. Then we went in for protein ligand interaction studies. We wanted to see whether the substrate for the polyketide synthase is really actually having any binding affinity towards the binding sites of this gene. And we did a Docky interaction and with the ferulyl CoA, which is actually the substrate of polyketide synthase. We found that there is a good binding of this ferulyl CoA, and the binding of the CLPK11 active site with ferulyl CoA was much more than the candidate, already known reported candidate gene, that is curcumin synthase 1. And we studied the intron-length polymorphism. We found that a little bit of polymorphism exists between the intronic region. Actually, within the coding sequence, if you evaluate, there is not much difference between the varieties, but within the introns, we can find some differences. And this intron length, can be also used as an identification for identifying the varieties. Then coming to the at this point of time, we thought it was needed to mine the upstream sequences of this gene, because definitely we expected that there is some change, some difference in the upstream sequence, which is responsible for binding of these regulatory molecules and regulation of this gene. So that's why we went in for the promoter sequence mining. We identified the upstream sequences, we characterized the sequences, and we found, surprisingly, we found 44 R2R3 MIB binding sites in this region, which clearly states that this gene is regulated by MIB transition factors. But we do not know the regulatory mechanism, whether it is a MIB alone that is regulating, or whether it is a complex involving MIB, DHLH, and WRKY factors. That we are not sure. So we are in the research. We are doing some research in that line also to identify the actual mechanism of this regulation. And these are the upstream sequences, and these motifs we have arranged. And see, this is one MIB, that is MIB 108 and MIB 4, that showed clear-cut binding to these upstream regulatory sequences. And so we got interested in these MIB transcription factors, and we went in detailed analysis of MIB transcription factors and identified and isolated and shortlisted 20 MIB transcription factors. And we did a differential expression analysis of one of these MIB transcription factors with our BAIT gene. We found that whenever this expression of this BAIT gene was downregulated, this MIB transcription factor was found to be upregulated. Means they have some negative correlation. Probably this gene was negatively regulated by this MIB transcription factor. We are yet to prove this hypothesis. And these are the number of, I told you, like 20 MIB transcription factors we have isolated from the transcriptome data. And we studied their physicochemical properties, predicted their structures, protein structure, hierarchical protein structure, and functional prediction we did. And then some of these MIBs we are now actually cloning their coding regions and actually characterizing them to see whether they have the the conserved domains and the typical characteristics like the conserved motifs and the subfamilial characteristics of MIB transition factors. And we found that all these MIBs, what we have identified, they indeed their MIB transition factors are indeed belong to this large class of MIB family. And this docking studies indicated that there is an interaction of this putative MIB with the promoter regions of our candidate gene. And we then studied the relative expression analysis of these different isoforms. We also found that MIB transition factors do exist as isoforms. And we found that some of the isoforms were correlating with the expression, while some were negatively correlated with the expression of the candidate gene. So this is actually, these experiments were done involving different tissues, different developmental stages, and contrasting genotypes. And everywhere we try to establish the co-expression and the co-regulation of these genes with the pathogens. And we have studied the tissue-specific expression. We have studied it in different accessions. Then under stress, without stress, everywhere we found that certain MIBs correlated, showed a positive correlation, while certain MIBs showed a negative correlation. So now it is a challenge to identify what is the actual regulatory mechanism behind regulation of these genes. Interestingly, we found that we have identified 2 isoforms for an R2R3 MIB and showing antagonistic effect. And we also, through docking, we predicted the binding residues of these transcripts. And right now we are trying, we are developing expression vectors, and then we are trying to do the transformation, genetic transformation, agrobacterium mediated genetic transformation for the validation of this, of our results. Because definitely you need to go for mutation studies to confirm your hypothesis. Now, again, we also did a protein expression study using iTrack, and all these pathogens, in fact, we could actually confirm through our protein data also. And in black pepper also, we identified the genes responsible for biosynthesis of piperine. Piperine is the most important secondary metabolite in black pepper. So this is a set of 19 genes which we had identified in the piperine biosynthesis pathway. Already these genes are known, but we found several gaps in the study, and now we have located these genes onto the chromosomes. About 26 chromosomes we have located the genes, and this is just, I wanted to show you a sample fragment of phenylalanine ammoniase from the whole genome. And we have mapped these genes onto the Piper nigrum chromosomes. You can see the distribution on the chromosomes. And several gaps were identified in this, and we are now looking for the biosynthetic gene clusters involved in both piperine biosynthesis as well as curcumin biosynthesis. But we are waiting for the full genome sequence data from turmeric so that we can, we can expand our study. Then next application is the cloning of a nutrient transporter gene. Again, I have gone back to the same transcriptome data I have been using for so many years. So from what I want to tell you is that if you develop a proper transcriptome, you can actually do a lot of things. Myriad number of applications are there from a single transcriptome, a properly developed transcriptome. So you have to plan very well before developing a transcriptome. This transcriptome can be used for, actually it can, the whole carrier you can use this transcriptome, and you can even give it to your successors. You can transfer this data. But only thing is that you have to do it appropriately. Your experiment has to be planned properly. Otherwise, the transcriptome data will be of no use to you. So now let me come to the next, how we have identified a novel gene responsible for nutrient transport. Actually, nutrient transport is very important. I am just showing you one example on zinc transporter, how we have mined it. We went in for, based on that from the transcriptome, that is a nutrient stress transcriptome, we identified certain zinc transporter genes showing differential expression between the stress as well as the control condition. And this expression analysis, we again by qPCR, we have confirmed that it was related to the availability of zinc. We actually used a zinc solubilizing bacterial strain, and then we applied different levels of zinc oxide which is sparingly soluble, so it is not available in the medium. And we analyzed the zinc in the basal tissue as well as in the medium, and then we found that whenever there is a zinc deficiency in the plant, the zinc transporter gene expression was upregulated. So we identified this particular transcript, and then we did a phylogenetic analysis, and we found that this zinc transporter was closely related to those present in another member of the family Zingiberacea, that is Bimsarculator. So we did an interpro scan analysis, and then we identified all the signature sequences in the transcriptome. in the novel gene, like the transmembrane helix, we found 8 strong transmembrane helices, which is a typical of a ZIP transporter gene. Then N-terminal signal sequences, then histidine-rich amino acid residues, and then N-methylation site, which all confirm that it is a zinc transporter gene. And then multiple sequence alignment was done, secondary structure prediction, 3D structure model, and then we also analyzed the promoter region and identified the zinc deficiency responsive elements present in the promoter regions, which are upregulated by the low zinc condition. But these genes are not present in the zinc deficiency. They are not zinc deficiency inducible. So that is what, this is a sub-DRE motif, 10 base per motif. And all these actually this it has come as a cover page. Our article has come as a cover page in Plant Molecular Biology Reporters. Some of the papers are under review, some have been published. Only published papers I'm showing you here. Then next application is the small RNA deep sequencing in turmeric by NGS. Basically, since I'm interested in the secondary metabolic pathways, all my work, basic aim is to identify genes and regulatory molecules involved in this biosynthesis. So coming to the small RNA sequencing, I think I have to move fast. So we have done deep transcriptomes, and then we have identified novel as well as conserved microRNAs. About 40 families of non-conserved microRNAs we have identified using this NGS-based deep sequencing strategy. And these novel microRNAs, see, these are the novel microRNAs we have. A few of them actually. We have followed the criteria of Schwab, certain stringent criteria was followed because our MIR base doesn't have any pre-deposited data is not available. And we have also gone in for identification of targets of these microRNAs. And yeah. So 97, I'll just go on to the summary. 97 novel microRNAs and 67 families belonging to 67 families were identified by domain-based sequencing. Okay. And 8 novel microRNAs we had actually validated by stem loop RT-PCR. This is how we have cloned and conserved microRNAs by stem loop RT-PCR. Primer designing in stem loop RT is slightly tricky. Many of you must be knowing. Then we have confirmed this by even by Northern blot analysis. These RNAs validated these RNAs by Northern blotting. And interestingly, we also found some human targets from these microRNAs, and that we do not know why we got human targets. We are yet to explore. And these results are also published. I think, yes, now I have come to the end of my presentation. Thank you for your patient listening. If you have any doubts, you can ask me. And last slide, I would like to show my team. The team involved, my students actually. My students and my mentor. Yes. Are actually involved in this work. Thank you.
Thank you so much for this insightful session, ma'am. We were really honored to hear this talk from you. There are a few questions that we have got in chat. I would like to share them with you. First is, okay, first is from Dr. Sumana Banerjee. They're asking, she wants to know how can you validate the marker trait association in curcuma, any deletion lines?
No, we don't have actually now, we don't have deletion lines. We are doing it now. We are trying to develop these mutants. Validation part we have not done, that I already told, we are working on it.
Okay. I hope it answers your question, Dr. Shomana. We have one more question coming from Dr. Malinath. They're asking, nanopore sequencing is increasingly getting popular. Why not use it?
Yes, I think we have nanopore little bit costly. Yeah, actually we are doing this whole genome resequencing now in black pepper. We've already initiated that work. And nanopore sequencing, whatever crop I am working on, I think in black pepper they have already done nanopore sequencing. That data is available. Whole genome sequencing is based on nanopore sequencing platform. And in turmeric, once funding is available, we'll definitely go for it.
Okay. Then Dr. Sri Kanta Prasad is asking, Madam, you talk about nutrient stress in relation with curcumin content. May I know which nutrients play a major role?
Actually, that question is not clear to me. Can you please repeat?
Yeah, I'm sure I'll repeat it.
Which nutrient plays zinc? Actually, I have worked on zinc. So, yeah.
I hope, Dr. Shrikant Prasad, you got your answer. In case if you want further explanation,
I have worked only on zinc.
If you want a further explanation, kindly elaborate your question a bit.
I think I am not the person to explain on nutrient things. So I can, if he can mail me that question, I can. There are scientists in my institute who can better answer this question on this nutrient.
Sure. Sure, ma'am. Same, Dr. Shrikanta Prasad is asking, ma'am, have you found any relationship with soil pH and curcumin biosynthesis genes?
Yes, definitely, there is a relation. See, and these genes are all very sensitive to environmental as well as growth conditions. So definitely soil pH also. We have to explore, but I'm sure that soil pH is definitely going to affect this biosynthesis.
Okay. Any more questions, guys? Please feel free to drop them in the chat section. I think we are pretty much done with all the questions. Thank you. So I hope there are no more questions that the participants have to ask from Dr. Shija.
Thank you.
Thank you so much, ma'am. Thank you for your time. Thank you so
Okay.
much. It was a really informative session, ma'am.
Okay, thank you.
So this pretty much brings us to the end of. Okay, ma'am. We just now got a question from Dr. Badaru. They're asking, can you list a few articles to refer based on this study?
No, exactly what, which topic? Biosynthesis?
Maybe, yes.
Biosynthesis of secondary metabolites?
Yes, yes.
Can you share me the mail ID of this particular
Sure.
person? I can send it. I cannot refer one or two. I have several articles. I can share with you all the references.
Ma'am,
I share his mail ID.
I'll do that.
Yeah.
I have dropped it as a private message.
Okay.
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 Any more questions, guys? Okay, I hope we are done for now. So, thank you so much, ma'am. Thank you for taking this
Thank you.
question.
Thank
you. This brings us to the end of day 2 of the webinar. Today's session was graced by talks from Dr. S. Paulraj, who talked about the utilization of sequencing-based approaches for deciphering rhizosphere microbiome for the sustainable crop production, and by Dr. T. E. Sija, who talked about utilizing NGS-based applications for mining genes and transcriptome factors involved in biosynthesis and regulation of important secondary metabolites. She also discussed in depth about utilizing transcriptome and small RNA-based sequencing approaches to achieve the same. Also, we had a talk on how a specialized and optimized computational platform like Genome Station and a dedicated genomics cloud storage platform called GStack can prove to be helpful to researchers for in planning their next analytics project. So with this, I would like to conclude today's session. A big thank you to all the speakers who managed to take time out of their busy schedules and share their insights with all the participants. My humble thanks to all the participants as well for sitting through the entire webinar patiently, putting across all their queries, and making sure that this webinar ended up being an interactive session. I would request all of you to join us tomorrow for the last and final day of the workshop. We have some interesting talks lined up for you. tomorrow as well. So we'll be more than happy to have you with us. Also, I am attaching the link of the feedback form for today's session. I would request all of you to just go through it and fill it, as your feedback will help us to improve. Thank you so much, guys. Thank you once again to all the speakers and all the participants. I would end today's session. Thank you so much.